Okay, now, the problem is this, that this system has 100,000 parameters for Clash, and Clash is a relatively small network. If we did it with target scan, there's a factor 10 in front of this number, right? So how do we fix these parameters, right? Now, so when I'm confronted, and also there's the thing that referees always ask about the values of parameters. I don't know what's wrong with them. I mean, they always ask about them. So, what do you do with this, right? So we just didn't do it, okay? So what we did is the following. We didn't try to find realistic values for the 100,000 parameters, okay? We tried to focus on the properties that are parameter independent. So those that are just generated, those just emerge with probability 1 for any realization of the parameters, okay? Those that in physics are called typical properties, right? And that in biology with some, I'm not 100% happy about this, but some people call them context-independent properties, okay? But ultimately, we put everything on a computer and we solved it and we looked at the matrices that came out and we looked at a bunch of properties there. So I'm going to focus on a few of them. I have no idea how I'm doing with the time. Can I?
Continue, I will tell you.
You will tell me when? Like when I have 1 minute?
Go, no problem.
Okay. So, let me try to, okay, so what we did in practice is this. We wanted to play with different parameters, but in a thermodynamic way. So we want to move one parameter at a time and keep all others fixed, but at the same time we want to see how things change in different scenarios, right? So there's two big scenarios that I want to focus on this talk, okay? One set of scenarios concerns the diversity of the binding sites. So we basically moved from a scenario in which there is the same kind of diversity that you see in the data, so this is just the distribution of the types of binding sites that you have there, to a scenario in which there's no diversity at all. All binding sites are exactly the same in terms of binding affinity, of strength, okay? And the second heterogeneity parameter we played with is the transcriptional heterogeneity of RNAs, both microRNAs and RNAs, okay? And basically we played with the relative variability of the transcription rate. The transcription rates are assumed to be random variables that we sample in each realization, right? And they have a certain variability, certain relative fluctuations. And they're drawn from log normal distributions where we just change the CV. So we move from distributions that have very little noise, so like this one here, the light blue one, to ones that are extremely noisy, so like the dark blue one here. Okay, so we just took the system, we sampled parameters in this way. This gave us binding strengths, this gave us transcription rates. It turns out that they are sufficient to recover all other parameters. Okay, all other parameters basically can be written as functions of these parameters here. So that's, once we started sampling from this distribution, we had our networks, and we could compute the susceptibilities, right? Okay. The benchmark is this. The only parameter that we move is the mean level of microRNAs in the network. So we just look at what happens when we pump more and more microRNAs on average in the network. Okay? So this is what you see on the x-axis here. And what we plot here are these two quantifiers that I think are relatively intuitive. So one is just the mean value of the susceptibility, averaged over realizations and over RNA pairs. Okay? And you see that that's a number of order 10 to minus 4. Again, susceptibilities don't have units, so this is good enough. And then the maximum susceptibilities that can be achieved across distinct IJ pairs, so not self-susceptibilities, but actual susceptibilities between different RNA species, are plotted down here. And this is a number that is about 4 orders of magnitude larger than this, okay? So on average, there's very, very little crosstalk in this network in terms of intensity, right? And it only happens in a certain region of values of microRNAs, so in this range here. On the other hand, there are very strongly interacting RNA pairs, very strongly cross-talking pairs. And this value is probably easier to benchmark against the value of the self-susceptibility, so how I respond to myself, essentially. And the value of the self-susceptibility is, because of how we define susceptibilities here, is 1. Okay, that's the maximum that you can achieve. So that's actually of the same order of self-susceptibilities. So they're pretty strong. Then the different columns here represent the different binding heterogeneity scenarios. So you see that the baseline crosstalk level doesn't depend at all on how you pick binding sites. While the maximum susceptibilities does depend somewhat on how you pick binding sites, right? You see that the peaks get generically higher, they tend to shift a little bit like this. But the most striking thing is how this depends on transcriptional heterogeneity, so on how diverse are transcription rates for RNAs. And they actually they tend to increase the level of crosstalk, of significant crosstalk, quite dramatically. Okay.
Yeah. The IJ pair is constant upon increasing beta. So the IJ pair that achieved the maximum success.
No. There is so
it's chaotic in IJ.
No. It's stable for a while. Well, I'm not going to talk about this, so you have to trust me. It's stable for a while, and then after a while you have some rearrangements, and then it's again stable for a while, and then there are some rearrangements.
Okay,
so it's not stable across the entire regime, but
it's
sometimes
stable.
Yeah.
Okay.
But the nice thing is that the strongly interacting RNA pairs are very conserved. So maybe you find a pair that is 10th in the ranking here, and 7th in the ranking here, and I don't know, 20th in the ranking here, but they're always in the top. So there's lots of conservation there.
Sorry, something I'm not sure I understand is the topology of the network that you are assuming is a given one based on I don't know, CMAP, MCF7 transcriptional network with microRNAs, or how are you? What is the topology of
the topology of the network? Well, it's taken from the data. So the data. The clash data. No, the clash data. The target scan data are different. So we use the clash data. So we really have a map of which RNA is bound by which microRNAs, and we keep that. So the topology is frozen.
And you have there also the interaction between the, and this is only transcription factors, right? We are talking about the transcription.
It's only messenger RNAs on one side and microRNAs on the other side.
Okay, and we have the transcriptional network on top of it, on top of the microRNA network?
No.
Okay.
It's just...
Would it be important at all? Because in a way, genes interact with genes.
That could be. But I think it's also a matter of time scales, right? So, but yeah, that could be.
Right.
That could be.
And what you do when several microRNA targets the same 3' UTR? Okay, you just sum.
I just compute it, and then I just look at the...
You sum the individual susceptibility, the individual...
No, the final susceptibility that I get between an RNA pair depends on all the microRNAs that connect them. Not only at a distance 1, but also far away along the network, right? So it's everything is in there. All right, so something a little bit more interesting is this. We try to understand whether, actually, this was the only thing I wanted to understand in this whole thing. That's the only thing. Everything else was kind of a side thing. So how much do susceptibilities between RNAs correlate with local interaction parameters? So with, for example, binding affinities, okay? So if you observe a high degree of correlation between the chi-IJs and the local interaction parameters, so binding affinities, then this means that crosstalk is mostly local. So the two RNAs that are crosstalking will mostly be separated by one microRNA species. But if the level of correlation is low, most crosstalking pairs will be far away, will be distant across the transcript network, okay? So this means that somehow the perturbation that I do here has to be broadcast all over the network in order for this guy to respond. So that's something not intuitive in the beginning. It's kind of a collective effect, if you like. And what I'm plotting here is the Pearson coefficient between susceptibilities and a parameter that basically quantifies how strongly they're interacting locally. Okay, as a function of the transcriptional variability here, and as a function of the binding variability here, and what you see is that essentially as you increase the level of transcriptional heterogeneity, this correlation goes down dramatically. So it's not very high in general. So this is a Pearson correlation, so it's not particularly high. There's not a lot of correlation with local parameters, but when there's lots of heterogeneity, this drops down. So most of the crosstalk is actually non-local, okay? Now I find this interesting for two reasons. One is that, well, let me tell you one for the moment. And many people try to predict crosstalk patterns based on local interaction parameters, and this says that it's not very good. It's not a very good idea, okay? And then the other picture that this suggests is that, of course, long-range interactions are important. So, for example, in a network like this, where you have microRNAs here, the red ones, and the green ones are RNAs, right? You will always have the microRNAs that is trying to silence somebody, right? So this guy who is next to him will be silenced, okay? But still, every time something like this happens, there's always a possibility that somebody broadcasts a perturbation here, and it goes all the way to the other side of the network, okay? We also looked at the typical distance of crosstalk interaction, and it's basically the diameter of the network. So they can be broadcast all over. Right. How am I doing with time? Sorry, I'm stressed out for time.
6 minutes.
6 minutes, okay. So what will I show you? Okay, let me try to compress a couple more results in this, okay? One of the other things that you can ask is, of course, because I have variability in the transcription rates, this will induce a variability in the RNA levels, right? And I can ask myself, how does the variability in RNA levels crosstalk with the crosstalk network? So how does the crosstalk network affect the variability in RNA levels, okay? And basically the net result is what you see in this plot, where the blue line represents, let's say, a normalized intensity of crosstalk, while the red line represents a normalized variability index, so a normalized coefficient of variation, just so that they can fit in the same plot, essentially. And what you see is that basically when you have stronger crosstalk, the fluctuations in RNA levels that are induced by the variability in transcription rates is actually suppressed. And as soon as crosstalk goes down, the variability increases. So there seems to be a kind of trade-off between these two things, okay? Another interesting aspect is the following. So we try to understand how the topology of the network is, what's the role of the topology of the network, right? So one thing we did is we basically looked at how different the crosstalk pattern, the crosstalk network that is generated in the actual clash, from the clash data is, compared to the one that you find in a edge-swapped clash network. So in a network that we generated by simply swapping edges in the clash network, okay? This is not much of a randomization, actually, because you preserve node degrees, okay? You destroy all order 2 statistics in the network, but node degrees are preserved. And you see that actually nothing changes. So this seems to be really encoded in node degrees. So there's something there, right? And if I destroy node degrees as well, then basically the crosstalk patterns that I find in the clash network and in the random network where I destroy node degrees is completely different, okay? And actually, some more details would be needed here, but the kind of crosstalk network that you find using target scan interactions is actually very, very similar to the one you get in a totally random network. And I think the reason is that in target scanning, the one that was available when we did this paper, they only included sequence-specific interactions. So now we really want to do the whole thing with the complete target scan with non-specific interaction, and I believe we will go back to the clash data. Okay. Okay, sorry, in the interest of time, I will just skip